every disease you study, there's a hidden world of enzymes behind the scenes, performing thousands of chemical reactions that keep the body and the disease moving. Enzymes are mostly proteins, sometimes RNA, that catalyze biochemical reactions. They speed up reactions by lowering the activation energy and aren't used up in the process, so they can work over and over again. Enzymes are classified into 6 major classes according to the type of chemical reaction they catalyze. Oxidoreductases are enzymes that catalyze electron transfer reactions, also known as oxidation-reduction reactions. Examples include lactate dehydrogenase, hydroxybutyrate dehydrogenase, glutamate dehydrogenase, and malate dehydrogenase. Because these enzymes are mostly inside cells, their serum levels rise when cells are damaged, such as during a heart attack or liver injury. As you can see in the picture, the oxidoreductase enzyme is in the middle, acting like a helper. On the left, molecule A is oxidized. It loses hydrogen ions and electrons, so it looks a bit sad. The enzyme takes those electrons and protons and passes them to molecule B on the right. Molecule B gains them, so it becomes reduced and looks happy. In short, oxidoreductase enzymes help move electrons and hydrogen from one molecule to another. One loses, one gains. Transferases are enzymes that move functional groups, like amino, phosphate, or methyl groups, from one molecule to another. Examples include AST and ALT, which transfer amino groups, and CK, which transfers phosphate groups. Clinically, AST and ALT rise in liver injury, while CK increases during muscle damage or a heart attack. As you can see in the picture, the transferase enzyme is in the middle, acting like a friendly helper. Molecule A on the left is the donor. It has a functional group attached to it. The transferase enzyme takes that group and transfers it to molecule B on the right, which is the acceptor. Molecule A loses the group, and molecule B gains it. In short, transferase enzymes move functional groups from one molecule to another. Hydrolases are enzymes that break chemical bonds by adding water, a process called hydrolysis. Examples include ALP from the liver and bone, ACP from the prostate and bone, amylase and lipase from the pancreas, and cholinesterase from the liver and red blood cells. Clinically, elevated ALP points to liver or bone disease. High amylase or lipase suggests pancreatitis, and elevated ACP can indicate prostate disease. As you can see in the picture, the hydrolase enzyme is in the middle, helping to break a molecule apart. Water is used in the process, shown by the little water drop. The enzyme uses water to break the bond in molecule A, splitting it into smaller molecules. After the bond is broken, molecule B is formed. In short, hydrolase enzymes break chemical bonds using water. Lyases are enzymes that break chemical bonds without using water or ATP, often creating a double bond or a ring structure in the process. A classic example is aldolase, a key enzyme in glycolysis. As you can see in the picture, the Lyase enzyme is in the middle, helping to break a molecule apart. Molecule A is broken without using water or ATP. When the bond breaks, parts of the molecule are released and a new double bond is formed. On the other side, the enzyme can also help form a bond by removing a group. In short, Lyase enzymes break or form bonds without using water or energy. Isomerases are enzymes that rearrange atoms within a molecule, forming isomers without adding or removing any atoms. A classic example is glucose phosphate isomerase. As you can see in the picture, the isomerase enzyme is in the middle, helping the molecule change its shape. Molecule A and molecule B have the same atoms, but they are arranged differently. The enzyme rearranges the structure of the molecule without adding or removing anything. In short, isomerase enzymes change the shape or arrangement of a molecule to form its isomer. Ligases are enzymes that join two molecules together, using ATP or another nucleoside triphosphate as an energy source. A common example is glutathione synthase. As you can see in the picture, the Ligase enzyme is in the middle, helping to join two molecules together. Molecule A is combined with another molecule to form a bigger molecule. This process uses energy from ATP, which helps create the new bond. In short, Ligase enzymes use energy to join molecules together.